You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you've enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you. Hello, this is Richard Jacobs with the Future Tech and Future Tech Podcast, Future Tech Health Podcast. My guest today is Martin Giera. He is an associate professor at Leiden University. We're going to be talking about his research into clinical metabolomics and some other stuff he does. So, Martin, thank you for coming.
Oh, welcome. I appreciate it.
Yeah, it sounds a little bit technical what you work on. So, can you break it down? Basically, what's the focus of your research?
It actually is, and that's a question many people ask. First, what is metabolomics? What are metabolites? Metabolites are small molecules. There's tens of thousands of them in the human body, active ones, inactive ones, storage components, structural components. And metabolomics actually tries to get an overview of all of them and their interconnectivity and how they are intertwined and steer and take part in biochemical processes. Obviously, with this knowledge, we hope to get a deeper understanding of human physiology and subsequently also about pathophysiological processes like specific diseases, which on the long run should help us deciphering novel treatment opportunities as well as advanced diagnostic approaches for many diseases where early detection is crucial. Just think about lung cancer or cancer in general, or diseases where we just don't know at all what is happening, like in amyotrophic lateral sclerosis and alike. And in my group, we use Future Tech to carry out these analyses.
So what is your focus on studying normal processes in the human body and what the metabolites are, or are you focused on studying certain diseases and what those metabolites are? What is your focus? Because it sounds like, with tens of thousands of compounds, you can't look at everything, obviously.
That's a very excellent point, and I think when metabolomics started, everybody was a bit too enthusiastic and had too high hopes, so to say, and we thought we could cover it all before we realized it's so many compounds. We cannot cover them all. So what is happening at the moment is that actually a lot of researchers around the world are taking apart the active ones from the non-active ones. So activity metabolomics, so where you profile the active metabolites, which are, so to say, the salt in the soup, they are in the focus, and they're also in the focus of my group. So it does not really separate into physiology and disease, but it more separates into active components versus inactive components.
When is something called a precursor versus a metabolite? Because like for certain processes in the body, I would guess there's not just one level of metabolites. Like you take in something and you chemically change it. So you had one thing and now you change it. Now you have a metabolite of that. But maybe that's an input for the next step in the cycle, the next step, and the next step. So how do you differentiate that?
Usually there's different levels of chemical modification, and these modifications can be mapped and analyzed. And on basis of these analyses, you can trace back what was first and what came later. And you can also have a look at the key players within these processes. And basically, when something leaves your body through urine or the feces, then obviously the story ended, so to say, for this metabolite.
So is a metabolite only when it finally is excreted from your body? I mean, an excretion, it sounds like, can be through your breath. Carbon dioxide, I guess you could consider an end-stage metabolite, right?
Yeah,
And
exactly. Yeah, correct.
sweat, yeah, sweat, urine, feces, I guess
Whatever is
that's it.
in there in small molecule content, creatinine, glucose, glucuronic acid, lactic acid, pyruvate. There's thousands, tens of thousands of them. I mean,
So what's an
by
example
definition, a
of
metabolite would be a product of an enzymatic reaction. So,
okay,
uh,
okay.
yeah, that would be
So
one of the basic definitions.
what's an example of a really fascinating pathway in the body and metabolites that you've discovered or researched, that you want to talk about? Any specifics?
I think what for me personally, what was a change in my career was when I visited the group of Charlie Serhan at Harvard Medical School, and he's looking at the metabolites of polyunsaturated fatty acids. You know them as the fish oils, and there's a big debate about it whether they are really beneficial or they are not really beneficial. And Charlie Serhan basically discovered a couple of metabolites of these unsaturated fatty acids, which become metabolized or modified within our body, and then they change in function and they become much more biologically active. And that founded the field of resolution physiology. So the understanding how inflammation in the body is actually actively turned off. I find this a very fascinating field where we also are engaged in many different projects.
So earlier on, when you said a metabolite is either active or inactive, you mean biologically active?
Yeah.
Okay. So um, will people excrete, by the time someone's ready to excrete something, again, through breath or urine or whatever, is the metabolite always inactive, or do people excrete active metabolites too?
Yeah, I guess that's a very individual problem or case for each and every single metabolite. See, if something is extremely active in your body, it usually undergoes a very rapid turnover. So it is made at the site of the action where it's needed, but it immediately is also metabolized further into an inactive form. Otherwise, the reaction of your body would be too tremendous and it would overwhelm the whole system. An example for such a metabolite is leukotriene B4, which is a very strong chemokine attracting white blood cells to the site of inflammation. You need it because you need the white blood cells to attack intruders like bacteria or uh, other organisms to clean it up. But if this production would continue and this metabolite would be very stable and linger around in your system, everything would get out of control. So at the very same moment, this very active metabolite is being further transformed into, in this case, 20-hydroxy leukotriene B4, which is way less active. So it's a very, it's you can see it like interwinded gears which work extremely nicely together with each other, and there's a very specific fine tuning occurring in the body, and this is also very fascinating if you think about it like this.
Yeah, so I was thinking of metabolites just as you eat food or you drink something and then you produce metabolites and it's used for energy production. But what are, I don't know, I know you can't define them all, but what are a lot of the major uses of metabolites in the body?
Oh, well, that's. I mean, we also say that the metabolome, so the entity of all metabolites, is the closest link to the phenotype. So basically, the metabolome largely defines a phenotype. So in this way, the metabolome also defines our physiology and can also define or cause diseases. I mean, not for nothing, we have the disease called metabolic syndrome, which basically is metabolism getting out of control, like too much food intake, too much sugars, too much fat. But there's many processes where metabolites play a crucial role. For me personally, what I think is most interesting is the signaling function. So that you see that metabolites basically have bioactivity within the body, and they have a plethora of different functions. And these functions are tissue specific or cell specific. So the levels of control and the levels of intertwined processes, well, that's close to a miracle, you can say, if you do research in that field.
Yeah, it's amazingly complex.
Yeah, it's really amazingly complex.
Can you give me a couple anecdotes? What are a few things that you've worked on that amazed you, that surprised you, and you really enjoyed discovering, over the past 6 months or year or several years?
Yeah, I think what we are just working on at the moment, and we have a study which has been submitted to a journal, and it's in revision. I cannot name details about it out of this reason. But when I did my PhD, I was researching the distal cholesterol biosynthesis, so the last steps in which cholesterol, a very important metabolite, which everybody knows actually, but no one realizes that we have approximately 300 grams of it in our body, and it's tremendously important for the liquidity of our biomembranes and the function of our cells. And I was researching this last pathway in the cascade to make cholesterol. And 10 years ago, when I wrote down my thesis, my PhD thesis, I was always thinking to myself, we always see this pathway of cholesterol biosynthesis just as a mere production of cholesterol. And everybody thinks that cholesterol is kind of a bad metabolite because you have high cholesterol, and we link this to this is bad for you. And I always asked myself, so is there not more behind what are the activities of these other metabolites? Because I could not believe that that all these steps occur just to make one final product. I always thought that there is much more about these other metabolites in the cascade. And now, 10 years later, I mean, one story we just published in Nature, actually some colleagues of us, but we're co-authors on this manuscript, we published in Nature showing that if you inhibit this distal cholesterol biosynthesis at a specific point, you can enhance the remyelination of oligodendrocytes. That's cells which are very important in diseases like multiple sclerosis. And we now have another study where we can show that certain precursors of cholesterol have very important biological functions. And if you interfere at the right stages in this pathway, and you actually cause the accumulation of this bioactive metabolite, you can change disease processes. And well, this for me personally is a very nice anecdote, because 10 years ago I already kind of had it in the back of my head. Something is going on here, but I couldn't really name it, and it took me kind of 10 years to round up the story, if you want to say so.
Yeah, it's interesting. It's so complicated. Let's say, like the people take supplements, let's say for health,
Yeah.
And then it makes me wonder, at what stage of a chemical pathway is the supplement they're taking affecting that pathway? What if, I don't know, you were tired and you wanted to take an enzyme to help your energy production. And let's say there's 10 steps in that pathway, and you're taking an enzyme that is step 2. How would it be different if you took one that's at step 4 or 5 or 8, or you took multiple ones? What would it do to the pathway? And so I guess there's such
we don't really know too much about this. Yeah. And the problem is also that the microbiome is also in between in many cases. And I think this is another level of complexity which we just start to understand at present. That, well, basically something which you ingest can be metabolized by your microbiome, and it can become a totally different metabolite. Also correlating with your actual microbiome, so with the bacterial species who are in your guts and are in your guts and my guts. So this is very individual, and this can tremendously affect how food is metabolized within the guts and what bioactive metabolites are actually produced by the microbiome. And then, you see there it links again. And there again, we need this technology, and we need to take apart the active ones from the inactive ones to understand how can we, or which are the juicy ones, so to say, so which are the ones we want to accumulate in the body, and then find the right tweaks to actually do that.
I wonder if anyone takes all this into account when they're looking to design drugs, or they just look for the end stage result. Do you think
this is just starting? Actually, this
Hmm.
to take metabolomics into drug research is just a field which is just at the moment really skyrocketing. So it took, in my experience, it took big pharma a little bit. So they are usually, the big pharma is rather conservative. Let's be frank about it, and they like to pick major technology. And metabolomics soared over the years. It became major, and now it also is finding its entry into the big pharma companies. So they are really picking it up at the moment.
Yeah, it sounds like there's a lot more room for improvement. I mean, let's say I don't know, I'll pick Viagra. I don't know what that pathway is of, you know, sexual excitement, but
Yeah,
there's X number of steps, and they just chose that one step, and there's probably room in there to really tweak the process by looking at all the other steps and seeing what adding this or adding that or suppressing this would do, and it makes it
yeah.
really complicated. It's crazy.
It is, and also the off-target effects. I mean, you take one drug, but we do not know all the effects it has. An example is paracetamol. What is wait a second? Acetaminophen. That's how you guys call it in the U.S. Acetaminophen. We
Right.
still don't know, after this stuff is on the market for 50 years. We still don't exactly know how it works. And the same is,
Really?
yeah, really. There is no 100% clear mechanism how acetaminophen is, well, alleviating pain. The same counts for aspirin. There is, over the years, there have been so many reports about other effects and new effects and side effects of aspirin and different processes it's interacting with. So, you can imagine that for many, many drugs we already have registered, there's also interest in looking at these drugs one by one and seeing if you might not be able to decipher other diseases where you could use it for, based on the knowledge how it interacts with the metabolome. And I think
What's
this is
weird
also
is
an
that
upcoming market.
yeah, what's odd is that it seems like it would just be too slow for big pharma, for instance, to look at one compound after another because there's all these interactions too. It makes you wonder how medicine can work at all given the complexity.
Yeah. Well, that's a different. I mean, that's maybe the flip side of it, if you want to say so. Because you see, we gain more and more knowledge, but this also makes processes more and more complex. And with one answer come three questions. And I
Yeah.
think there you have to be very careful to which point you go, because you can easily get lost in these details and get lost in the complexity. And then in the end, you don't produce any new drugs anymore, anyways, because you just come up with more demands and more questions. So that's a good point. Yeah, you have to be careful there.
Well, I guess one good thing is if you look at the typical person, that there are hundreds of different reactions going on, or maybe thousands or tens of thousands, all kinds of metabolites being produced at once, and they don't seem to be interacting in a bad way because the person lives and thrives, at least for
Yeah,
its lifespan. So it seems like, I guess a good step would be looking at the human body and knowing, all right, at least it has figured out the rules where all these things can coexist peacefully. So I don't know, to try to do that in the lab just on your own, it seems like the human body itself, you don't want to experiment on people, obviously, without knowing what's going on. But it just seems like I guess, like a living creature, like a lab rat. That's why they're such a powerful vehicle
yeah,
to figure out what will work and what won't, because they have this whole rule book in the background running where things seem to work. But for us as scientists, or for you as a scientist, it's really hard to figure out all the factors that would go into making a pathway work or not, or influencing it. You know what I'm saying? I'm trying to
but
describe
that's
it, but
unfortunately, this is also the reason why we still need all these lab rats, so to say, all these test animals, because it's too complex to mimic it in a petri dish.
hmm.
I mean, also there is stupid things done with laboratory animals, which should be avoided on the other hand, you cannot just give something to a human being. You need to test it in a mouse or a rat, because we cannot mimic the complexity of all of this within one single petri dish. We're not at this level. And the other thing I think is that artificial intelligence and neural network computing and these type of novel approaches will revolutionize the field of metabolomics. Putting this, what you just brought up, putting this into context. So where is the equilibrium between all of these different players, and how are they exactly working together? And what happens if I tweak metabolite B? What happens to the whole system in a dynamic way? And I think only with artificial intelligence we can get a full picture of this, because it is already now at the stage where we are. It is very hard to grasp for us.
Is there a particular pathway that is like the one that's the most understood by science? Is there any examples of one that we think is well understood?
Cycle. Yeah, citric acid cycle, I would say.
Is that the Krebs cycle, the citric acid cycle?
Krebs cycle. Yeah, Krebs cycle.
Can you just give a few details about it? Like how many steps, how many intermediate things are produced? Any numbers on it?
Well, it's our central pathway for energy production. So all the ATP molecules, which is basically the fuel of our cells, is mainly produced within the Krebs cycle. It burns glucose, and by this we breathe. We excrete CO2, and we make energy to wrap it up. Now the steps, I would have to think about
Well, it's approximately
by
how many
heart,
steps
I would say it's maybe 10 steps. The interesting things is that it also plays a major role in cancer. Many people will know the Warburg effect, which was found by the Austrian physician Warburg somewhere in the 20th century, 1910 or something. And he found out that there is this paradox that cancer starts to ferment basically glucose. So you produce a lot of lactate, and with this you produce much less energy per molecule of fuel. If you would otherwise go through the whole Krebs cycle, you could produce much more energy for one molecule of glucose. And he found this paradox because, cancer cells, they grow much faster, so they need a lot of energy. So the paradox question is, why do cancer cells switch in their metabolism to a fermentive process? Why do they not use the entire citrate or Krebs cycle anymore? There's a lot of papers and studies about this, and a lot of research has been put into this, and it is still one of the very, very central cycles within cancer. And I think we start to understand more and more how this is put together. But this is only in total, maybe if you take glycolysis and the citric acid cycle together, there's maybe 10 steps or something. I don't know it now by heart. And this is already very complex, to measure all the different intermediates, to make heads and tails from where does stuff come from where does a metabolite enter it. There is more than one entry into these cycles. Trace back where does it come from. But the interesting thing then, if you do this type of research and you look at, and even if you look only at this snapshot, this very focused small area of the entire metabolism, you can see that when you profile different cancers, that they are all different metabolically. So it's a very individualized process, this metabolic process. And on one hand, this is of course a problem because one cancer is not like another cancer. But on the other hand, it kind of proves that we need personalized therapy for cancer. And it gives us the chance to, in a very specific way, possibly, interfere in a very individual way with different types of cancer and within different individuals. I think this is also where we're going to respect.
Yeah, I think it's probably a very good thing that cancers don't use the Krebs cycle in the traditional way, because now there's possibly a way to target them and to cut them off from their energy production.
I wouldn't say that, because on the other hand, No, I'm not sure if I recall it by heart correctly, but I think I remember that I read some papers that when you normalize it again, the malignancy goes down.
Huh.
So normalizing metabolism might actually also be an entry into lower malignancy. So that's a bit of the chicken and the egg question, right?
Yeah, right. That's true.
Can I change the chicken when I change the eggs? Or in other words, if I normalize metabolism, can I normalize my cell's behavior? Or is it the other way around, that the disturbed behavior of the cell causes the disturbed metabolism? But I think there is no clear answer to this yet. But this is a very active field of research as well.
Yes, I know we're close to being out of time. So what are you working on now and over the next 6 months or a year? Let's talk about that, and then we'll finish up.
Well, yeah, resolution of inflammation is still a hot topic. We're pursuing novel targets to activate the resolution of inflammation. We're doing that or trying to decipher those by using our metabolomics and lipidomics techniques. Cancer metabolism is also an active field where we have a lot of collaborations. Personalized medicine is coming up on the horizon as well. And one bigger thing where we're also active in, and this is also a bit to wrap up where the field at the moment is, we still need to set a good baseline for human physiology. So for many of the metabolites we can measure, we don't know what are the actual physiological ranges. And there we are engaging in a couple of collaborations with many colleagues to accomplish this and to give good numbers for the physiological ranges of our metabolism and our metabolites.
When you say physiological ranges, do you mean the concentrations or the amount of certain metabolites?
Exactly. So there is, for many of those, we don't have a clear-cut answer. So what is the range we can expect is one metabolite in?
Gotcha. Okay. Well, just like when you take a blood test and your, whatever marker is within a certain range.
Exactly.
I guess we've established supposedly that these ranges are healthy or not healthy, and then you have for glucose or for a couple of more, but now we're talking about hundreds to thousands of these parameters where we need to define the physiological range. Gotcha. Well, I guess to say you have a lot of work ahead of you is an understatement, but it sounds like really fascinating stuff, and I can see why you work on it. Yeah. Well, very good. Well, Martin, what's a good way for folks to get in contact, for collaboration or ideas or questions in case they want to reach you?
I think with my last name, you will always find me in the internet. There's only a couple with that name in the world. So if you just Google me, you'll find me and you have my email address and just drop me.
Okay. Well, very good. Well, Martin, I appreciate you taking the time and thank you for coming.
Yeah, I appreciate it too. And thanks for the invite.
You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you've enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you.